The deadliest plague in human history accidentally created humans who are immune to HIV. But here's what nobody talks about. If your ancestors survived the Black Death, there's a chance you're carrying something in your DNA right now that pharmaceutical companies have spent billions trying to replicate. In 1347, 12 ships pulled into the Sicilian port of Messina. The sailors on board were either dead or covered in black, oozing boils. Nobody knew it yet, but Europe had about 5 years left as they knew it. The bacterium responsible was Yersinia pestis. Nasty little thing. It attacked your lymphatic system, causing these golf ball-sized swellings called buboes, usually in your armpits, neck, or groin. They turned black. Then they burst. I'll spare you the details, but let's just say medieval medicine involved a lot of prayer and not much else. Here's where it gets insane. Within 5 years, 30 to 60% of Europe's entire population was dead. Gone. We're talking 25 to 50 million people in an era where news traveled by horse. The disease spread faster than warnings about it. Bodies stacked up so quickly that individual graves became a luxury. Mass burial pits, some holding thousands of corpses, became the only option. Gravediggers worked around the clock. Many of them died mid-shift. And the speed? Most victims went from healthy to dead in about a week, sometimes less. Families watched everyone they loved disappear before they could even process what was happening. Some villages lost every single resident. Not most, every. These ghost towns sat empty for centuries because nobody wanted to go near them. But here's the thing that doesn't get mentioned enough. Not everyone died. Some people nursed the sick, handled rotting corpses, practically bathed in plague bacteria, and never got sick at all. Medieval folks figured God was protecting them. Turns out the answer was written in their blood the whole time. And that blood, it's about to explain why some people today are basically immune to HIV. While entire families dropped dead in the same week, some people just didn't. And it wasn't because they were hiding in their basements eating garlic. These survivors walked through streets lined with corpses. They nursed dying relatives, held hands with the infected, even helped bury bodies in those massive plague pits, basically swimming in bacteria. Nothing happened to them. Medieval folks had their explanations. Divine protection, extra prayers. Maybe they ate the right combination of herbs. My personal favorite theory from the time? Bad smells caused the plague, so good smells would prevent it. People literally walked around sniffing flowers while their neighbors died. Spoiler, it didn't work. But here's what actually saved them. Their blood was different. Not in some mystical way, in a very specific, measurable, genetic way. Modern scientists figured this out by doing something slightly creepy, but incredibly useful. They dug up plague victims. Researchers extracted DNA from teeth found in those old burial pits. Teeth preserved genetic material surprisingly well, even after 700 years. What they found changed everything we thought about survival and luck. Certain gene variants showed up way more often in people who lived through the plague than in the general population before it hit. We're talking statistically significant differences, not coincidence, selection. Some families had survivors across multiple generations, grandparents, parents, children, all making it through while neighbors vanished. That's not prayer working overtime. That's inheritance. These lucky genetic lottery winners went on to have kids. Lots of kids, actually, since there was suddenly plenty of room in Europe. Their descendants spread across the continent. Which brings us to a tiny protein sitting on your immune cells right now. It's called CCR5, and it's about to explain everything. CCR5. Sounds like a droid from Star Wars, but it's actually a tiny protein sitting on your white blood cells right now. This little guy acts as a doorway, a loading dock, really. Certain germs pull up, grab this protein like a handle, and waltz right into your immune cells. Uninvited guests using your own front door. Now, CCR5 isn't some useless decoration. It helps regulate inflammation, coordinates immune responses, does actual work. Your body made it for good reasons. But here's the problem with having doors. Sometimes the wrong things know how to open them. This protein receptor shows up on T cells, macrophages, and dendritic cells. If those names mean nothing to you, just know they're the soldiers of your immune system. The very cells that should be fighting invaders, they're the ones with welcome signs on them. Scientists didn't even know CCR5 existed until the 1990s, and they only figured out it mattered because of a completely different mystery. See, researchers were studying HIV patients. Standard stuff, but they kept finding these weirdos. People who should have been infected, partners of HIV-positive individuals, healthcare workers with needle accidents, sex workers in high-risk areas. By every calculation, they should have caught the virus. They didn't. Their blood tests came back negative, again and again and again. At first, scientists assumed lab errors, then maybe lifestyle differences. Prayer, garlic, who knows? But when you see the same impossible result dozens of times, you stop calling it impossible. Something in their biology was different, something genetic. Turns out these protected people had a broken version of CCR5, not just slightly different, completely mangled. Their immune cells didn't have doors at all, and that broken version, it traces straight back to medieval Europe. Here's the thing about CCR5 delta 32. It's not a subtle mutation. It's not like changing one letter in a sentence. No, this is like someone took scissors to your DNA and just snipped 32 base pairs of genetic code. Gone. Vanished. Poof! That missing chunk creates a protein so mangled, so utterly broken, that it never even makes it to the cell surface. Your body tries to build a doorknob, but what comes out looks like modern art. Useless modern art. And here's where the numbers get wild. About 10% of Europeans carry one copy of this mutation. One copy. That gives you partial protection. But roughly 1%? They hit the genetic jackpot. 2 copies, complete protection. Their immune cells have no doors whatsoever. Now, I know what you're thinking. Doesn't that cause problems, living without CCR5? Surprisingly, no. Well, mostly no. People with 2 broken copies appear completely healthy. Other receptors pick up the slack. It's like losing your front door, but having a perfectly good back entrance. Inconvenient for some guests, but the house still works. But here's the really strange part. This mutation barely exists outside Europe. African populations, almost zero. Asian populations, basically none. It's like Europeans have a secret genetic password that nobody else got invited to the party for. Scientists initially thought the mutation was recent, maybe a few hundred years old. Then they started digging, literally. Ancient DNA analysis pushed the date back further and further and further. We're talking at least 2,900 years ago, long before any medieval plague showed up, so the mutation existed for millennia, just sitting there, waiting. Then the Black Death arrived, and suddenly, having broken doors became the difference between life and death. The Black Death didn't just kill people, it selected people. And there's a difference. In 2022, researchers published a study in Nature that made geneticists lose their minds. They analyzed DNA from over 500 individuals who lived before, during, and after the plague. Samples came from London, Denmark, and several other European locations. What they found was staggering. People carrying the CCR5 delta 32 mutation were up to 40% more likely to survive infection. 40%. That's not a minor advantage. That's the difference between flipping a coin and rolling loaded dice. Here's how brutal selection pressure actually works. Before the plague, maybe 5% of Europeans carried this mutation. After the plague, that number essentially doubled in just a few generations. Evolution usually moves like a glacier, slow, steady, barely noticeable within a human lifetime. But the Black Death? It hit fast forward on the remote. I think about this sometimes. My ancestors probably survived because of random genetic luck. Yours too, if you're European. We're all walking around with DNA shaped by the worst catastrophe in human history. The math is almost uncomfortable. If 60% of Europe died, then every single European alive today descends from the lucky 40%. The survivors weren't just fortunate, they were genetically different. And the wildest part? Most of them had no idea why they lived. They assumed God liked them better, or their prayers worked harder, or maybe they just ate more garlic. Nope. Their cells were just missing a doorknob. Natural selection compressed thousands of years of genetic change into roughly a century. The plague essentially speed ran evolution, but knowing this mutation helped people survive is one thing. Understanding exactly how it blocked the plague bacteria, that requires getting into the actual biology. So how did a missing protein actually stop a bacterial killer? Here's what scientists believe happened inside the body. Yersinia pestis wasn't just floating around in the blood. It was sneaky. The bacteria specifically targeted immune cells, macrophages, T cells, the very soldiers your body sends to fight infections. The plague turned your own army against you. And CCR5, it was the side entrance. The bacteria likely used this receptor, or something very similar, to slip inside immune cells undetected. Once inside, Yersinia pestis basically set up camp. It multiplied. It spread. It killed from within. But Delta 32 carriers? Their immune cells were missing that side door entirely. The bacteria knocked, but nobody answered. They still got infected, sure. The plague could find other ways in, but their immune systems bought precious extra time. Here's where it gets interesting. Many plague victims didn't actually die from the bacteria itself. They died from their own immune response going nuclear. Inflammation spiraled out of control. Organs failed. The body essentially attacked itself, trying to fight back. Researchers found something fascinating in lab studies. Macrophages without CCR5 don't just resist bacterial entry better. They also produce a more measured inflammatory response, less friendly fire, fewer casualties from the immune system's own weapons. It's like having a security team that stays calm under pressure instead of burning down the building to stop a burglar. The protection wasn't foolproof. Plenty of Delta 32 carriers still died. But in a disease where survival was basically a coin flip, having even 30 or 40% better odds meant everything. Your ancestors won a genetic lottery they didn't even know existed. But here's the truly wild part. 600 years later, a completely different pathogen would try using that exact same door. HIV showed up in 1983. Scientists knew it was destroying T cells. They just couldn't figure out how. For 13 years, researchers watched this virus slip past every defense. It was like watching a burglar break into houses, but never catching them at the door. Frustrating, doesn't even begin to cover it. Then, 1996 happened. A team finally cracked it. HIV wasn't picking one lock. It was picking two. The virus first grabbed onto CD4, a receptor everyone already knew about. But that wasn't enough. HIV needed a second handshake to get inside. Guess what that second receptor was? CCR5, the exact same protein our plague survivors were missing. Here's how the virus works. It latches onto CD4 first, like grabbing a doorframe for balance. This changes the virus's shape. Then it reaches for CCR5, uses it to pull itself flush against the cell membrane, and basically injects its genetic material straight inside. Without CCR5, the virus just dangles there uselessly, attached to CD4, but locked out. It's like having a key that fits the deadbolt, but not the chain lock. You're stuck in the hallway. This discovery explains something doctors had noticed for years. Certain people were basically bathing in HIV exposure. Partners of infected individuals, healthcare workers with needle sticks, high-risk groups with constant contact, yet some of them never got infected. Not once. Turned out many of these mysteriously immune individuals carried Delta 32. Their medieval ancestry was literally protecting them from a 20th century epidemic. 600 years apart. Two completely different pathogens. Same genetic backdoor. The math was almost too perfect to believe. But believing wasn't enough. Scientists needed proof that removing CCR5 could actually stop HIV cold. They were about to get it in the most dramatic way possible. Timothy Ray Brown was dying twice. HIV had been attacking his immune system for years. Then leukemia showed up, because apparently the universe has a sick sense of humor. Brown needed a bone marrow transplant. Standard procedure. Find a matching donor, wipe out the old immune system, rebuild with new cells. Doctors do this all the time. But his physician in Berlin had a crazy idea. What if they didn't just cure the leukemia? What if they cured the HIV too? See, Brown needed a new immune system anyway. His old one was getting destroyed regardless. So his doctor went hunting through the bone marrow registry for something specific. Not just any match. A match who carried 2 copies of Delta 32. He found one. In 2007, Brown received his transplant. New bone marrow, new immune cells, and crucially, cells with no functional CCR5 whatsoever. Then came the moment that changed everything. Brown stopped taking his HIV medication. Just stopped. Every doctor watching held their breath. HIV always comes back. Always. The virus hides in reservoirs throughout your body, waiting for its chance. Weeks passed, Nothing. Months passed, still nothing. Years passed. Brown's blood remained completely free of detectable HIV. The medical community lost its collective mind. 13 years. That's how long Brown lived HIV-free before cancer finally took him in 2020. Not AIDS, not complications from HIV, regular cancer. For over a decade, the virus that had been slowly killing him simply vanished. Other patients have since replicated his results. Same approach, same outcome. The transplant itself is dangerous, but it works. Scientists had their proof. Now they wanted something better, something that didn't require nearly killing someone first. Scientists watched the Berlin patient and had one thought: there has to be an easier way. Bone marrow transplants work, but they also nearly kill you first. The procedure destroys your entire immune system before rebuilding it. That's like burning down your house to get rid of termites. Enter CRISPR. This gene editing tool lets scientists cut DNA at precise locations, snip out those 32 base pairs, create the delta 32 mutation artificially. No bone marrow transplant required. The approach is surprisingly straightforward. Doctors remove a patient's T cells, edit out CCR5 in the lab, grow millions of these modified cells, inject them back. Early trials show real promise. Some patients saw their viral loads drop dramatically. Their edited cells survived and multiplied. The virus couldn't touch them. But here's where things got controversial. In 2018, a Chinese scientist named He Jiankui announced he'd created something unprecedented: twin baby girls, born with edited genes. CCR5 deliberately disabled before they ever took a breath. The scientific community exploded. Not with praise, with fury. Those babies didn't have HIV. They didn't need protection. He Jiankui had experimented on healthy embryos, permanently altering their genetic code and the code of every descendant they might have. He's currently in prison. The technology itself isn't the problem. It's the ethics. Editing sick adults who consent? Most scientists support careful trials. Editing babies who can't consent? That crosses lines we haven't finished drawing yet. Still, the potential remains staggering. What nature accomplished through 50 million deaths over centuries, medicine might replicate in a single procedure. A one-time treatment instead of daily pills costing thousands monthly. But Delta 32 isn't the perfect shield everyone assumed. Turns out the mutation that protected Europeans from plague comes with its own hidden price tag. Here's the twist nobody saw coming. CCR5 isn't junk. Your body actually uses it. People missing this protein seem to catch worse cases of West Nile virus, like significantly worse. Studies show Delta 32 carriers end up in hospitals more often when that particular mosquito-borne infection hits. Not ideal when you live somewhere with mosquitoes, which is basically everywhere. And influenza? Same story. That protein you don't need for HIV? Turns out it helps coordinate your immune response to the flu. Without it, your body's defense team shows up late to the fight. Evolution is basically a used car salesman. Sure, this mutation runs great against plague and HIV, but the transmission sticks sometimes, and don't ask about the air conditioning. There's no free lunch in biology. Every advantage comes packaged with a disadvantage somewhere. The ancestors who survived the Black Death passed down protection against one threat while potentially increasing vulnerability to others, a genetic trade-off written in their DNA. Now here's what really gets me. 40 million people live with HIV right now. Today, the mutation that could help most of them exists in less than 1% of the global population, and almost all of those people are European. The Black Death essentially ran a brutal genetics experiment on one continent. It killed millions to create survivors with a specific advantage. 7 centuries later, that advantage matters again for a completely different disease. But the protection isn't universal. It never was. Nature doesn't design perfect shields. It builds compromises. Survive this, risk that, win here, lose there. The plague's genetic legacy saved some lives and will cost others. That's not tragedy or triumph. That's just evolution doing what it always does, making deals with consequences we discover later.